Well, thank you very much for inviting me. I'm a computational neuroscientist, not a imaging engineer, but I'm one of the computational neuroscientists who's always believed that human vision and computer vision and imaging have a lot to teach each other, and that these two fields can walk hand in hand. And I think you'll see a lot of computer vision and image processing ideas sort of inspire my work. I'm interested in how the human brain sees, and I'm going to discuss this in 3 or 4 parts in the next 45 minutes. The first is an overview of the problem. Of course, the trouble is we've got this complicated brain here, and it's made up of a lot of different bits. There are the overlying cortex, that's sort of like the skin of a turkey, and then there are the inner parts like the thalamus and the lateral geniculate nucleus and all those subcortical nuclei that are kind of like the giblets of the turkey. And for us mammals, humans, most of the interesting computation that occurs on the surface of cortex. And primates, of course, humans included, are very visual animals. If you take a non-human primate like a macaque monkey, it's about 50% of its brain is vision. And for humans, it would be about 25% of the brain is visual. So it's a very important system for primates, and one that is hopefully accessible and one that we can maybe solve before the heat death of the universe. I tend to think of the brain as a computer. So on the left here, we have a standard Intel chip. And of course, this system is designed. Its functional units are known. They're transistors. You can sort of look on this chip and see that the transistors are organized in little banks, and you might get some idea of what the individual parts are. And the hardware and software, of course, in this system are independent. This is a universal computer. It can run any piece of software. On the right is the human brain. We have the complicated cortex, the cerebellum. This is an evolved system. Its functional units are unclear. We know that at the core of the human brain, there are about 10 or 11 billion neurons, but how these neurons are organized into local circuits and columns and areas, we really have very little idea at this point. And of course, the most important thing is that the hardware constrains the software, because on a short time scale, you can't grow a new synapse or a new neuron. It takes about 20 minutes. So that means that as you're sitting here thinking and your thoughts are switching from one thought to the next, you have to be doing this with essentially a fixed set of wires. And somehow or other, these wires modulate activity and information flow through the network to give rise to different thoughts. So if you're going to work on human vision, it's always a good idea to start with a model system that we know a lot about. This would be the macaque monkey. On the right here is a macaque. This is the lateral view, this is the medial view, and you can take the cortex and sort of inflate it and then flatten it out. And for a macaque, you would have something about the size of a large cookie. And then you can paint in the visual areas here. This is primary visual cortex and a bunch of other higher order visual areas. In the macaque, as of 1992, there were about 32 visual areas that were known. There's about 40 now. These are organized in a complicated network. Information comes in here at the retina, flows up through the circuit, up here to the highest point in this particular model, which is the hippocampus. But of course, it's flowing to all other sorts of parts of the brain, the frontal cortex that's involved in planning, the motor system, and so on. You can see that there's a lot of modules, and they're hooked together in a complicated network. It seems that the brain is about 50% interconnected at this point. So if you have, say, 40 visual areas, you could have 1,600 connections between those areas. And it looks like there are about 800 connections between those areas. So it's a very highly interconnected network, and there's extensive feedback. Every area that feeds into another area, there's also a feedback connection from that area. So this is a fairly complicated system. It's a nonlinear dynamical system whose operating principles at this point are not really well known. Generally, you can divide the network up into 3 levels. The early visual system, which is involved in representing very simple structural features like edges. The higher level visual system, which is involved in representing the semantic content of scenes, and also is involved in coordinate transformations to map information from the retina onto the motor system. And the mid-level areas that we think mediate grouping and segmentation, for example, figure ground organization. This is a critically important problem for human vision. It's also, as you probably know, a very difficult and important problem in computer vision. What we want to do in the human case is divide up these areas and figure out sort of what each area does. In other words, what each area represents, and how the system is organized into areas, and essentially what their computational role is. Now, until recently, we didn't have a good way to do this in humans, but in about 1993, people realized that there was a technology that existed called MRI that could be used to recover functional information about the brain. So MRI is, of course, a chemistry technique that goes back to World War II. And in about the 1970s, it was begun to be used to recover anatomical information from the human body. And in 1993, people realized that what had been considered to be sort of an artifact or a bug in MRI was actually a feature, and that MRI could be used to recover information about blood flow, blood oxygen, and blood volume in the brain. Now, neurons are essentially little engines. They're chemical engines. They make ATP, and they use it as their fuel. And to make ATP, they have to burn sugar with oxygen. So when neurons fire, there's a compensatory reaction in the brain that changes the flow of sugar and oxygen to regions of the brain that are quite active. And that is this indirect echo is what we measure in MRI. So MRI is not measuring neurons. When you see someone who does MRI say they're measuring neurons, you should correct them. It's not correct. It measures blood flow, and blood flow is only indirectly and nonlinearly coupled to neurons. But it turns out that since about 80% of the neurons in the brain are excitatory, that there's a general correspondence between changes in blood flow and changes in excitatory neural activity in the brain at a very vague level. So this is some typical MRI data. On the left, we have a movie that people are watching. These are not my children. These are far too cute to be my children. People are staring at a green spot here in the middle of the screen. On the right here is a brain, and we've divided the brain up here, you see, into little tiny squares. These squares are voxels. They're essentially volumetric pixels, and MRI is measuring things in voxels because it's a Fourier domain technique, and when you do the inverse Fourier transform, you end up with three-dimensional pixels. Now, the brain's inconveniently folded up inside the head, excuse me. So we unfold the brain computationally. We end up with a left and a right hemisphere. These are collectively about the size of a large pizza. And then we can zoom in onto one hemisphere, and we can overlay in white a bunch of the visual areas that we sort of know about. These are the visual areas back here. This is the motor and somatosensory strip. This is auditory cortex. These are some speech areas and some eye movement areas. And these little squares here are the voxels now distorted because we've unfolded the brain. And on here, the red regions indicate more blood flow, more blood oxygen use, and blue indicates less blood oxygen use. And you can see that this map is changing quite dynamically as people watch these movies. Sometimes there's more blood flow, sometimes there's less blood flow. In the visual system, of course, these changes are occurring most rapidly, as you would expect, because this is a movie. But you see changes even up here in prefrontal cortex, which is involved in planning and thinking about the future and moral judgments and so on. The pattern of activity across the map here is fairly complicated. And our problem as neuroscientists is to sort of just figure out what each bit of the brain does, right? We have essentially 60,000 points we've measured on this map, and we want to find out what information in the movies is driving activity in each one of these 60,000 points. Now, if we're measuring a point in the early visual system, say primary visual cortex, you might expect that that's going to be driven by a certain selection of pixels. But if we're measuring a point up here in higher visual cortex that's involved in the semantic qualitative properties of the scene, then there may not be any direct relationship between the pixels on the display and brain activity. It's going to be a lot harder for us to figure out what the brain is doing. But hopefully all of you can see that this is essentially just a system identification problem, classic system identification problem from engineering. I've got a black box, in this case a gray box, and I just need to figure out what this black box does, and I have to figure out what input will optimally give me the transfer function of the black box when I read the output. So in this case, the input is pixels, and the output is some activity space that I've measured from the brain, because of course I'm not measuring neurons, I'm measuring brain activity. And I just want to find some mathematical function that maps the pixels into the activity. And of course, if the brain was linear, this would be trivial. If it was nonlinear, we could do a piecewise linear approximation and kind of hack at it, and it would sort of work. But the brain's a nonlinear dynamical system, and system identification doesn't really work very well when we have a nonlinear dynamical system. In fact, if we have an arbitrary nonlinear dynamical system, all of you know that we actually can't solve that system. We can't guarantee that we can solve the system. So this means, unfortunately for me, that my career is kind of hopeless, but let's just ignore that for the time being and continue along pretending that we can actually solve this system. So since this is a system identification problem, there are a huge variety of methods we can use to solve this system, hundreds of different methods. This is one that happens to be fairly simple to think about because it conveniently divides the problem up into two parts, a sort of hypothesis-driven part and a modeling part. What we assume is that the relationship between the pixels and the activity space is nonlinear. Of course, it's nonlinear dynamical, but let's assume for the time being that it's stationary and has constant variance. So now, in that situation, we should always be able to find a feature space such that the mapping between the pixel space and the feature space is nonlinear, but the mapping between the feature space and the activity space is linear. And you know you can always do this because you can always stick a linear transformation at the end of any arbitrary system, and it doesn't change the system, right? So we can always find some feature space such that the relationship between the feature space and the activity space is linear. Now, this breaks the problem up into two parts. First of all, we have a hypothesis about how the pixels are transformed into some feature space and what features are represented at each point that we've measured. And then we can solve the mapping between the feature space and activity space with linear regression. And of course, linear regression is something we're really good at. We have a billion ways to do that. That's really easy. So this is an approach most often taken in my lab. We don't always do this. Sometimes we use more complicated methods. For example, you could solve this pixel to activity space transformation using a deep belief network. But then all you would have done is taken your hideously complicated, uninterpretable system from an in vitro system from an in vivo system to an in vitro system in your computer, and you would still have to figure out why it works. And as those of you who have used those kinds of neural network architectures know, it's very difficult to figure out why they're doing what they're doing. So now the problem is, for every point in the brain, we don't necessarily know what feature space that point in the brain is representing. So we need to compare several feature spaces that are essentially instantiated as different hypotheses. So we collect first a fit data set where we show some movies, and then we collect some brain activity data. And now we put the brain activity data to the side and ignore it for a moment. And we take our movies and we transform them into different feature spaces. So for example, we might compute motion energy in a giant Gabor wavelet motion energy pyramid. We might extract the semantic information in the scenes. We might do this in a hierarchy. There are semantic hierarchies from linguistics, for example, WordNet, that we could use for this. Or we might use some statistical algorithm. For example, we could use latent Dirichlet allocation to recover the latent scene category structure of the movies, and then maybe there's a part of the brain that represents those scene categories. So now we've move the movies into several different feature spaces. And now for every single voxel we measure in the brain, all 60,000 voxels, we use linear regression to fit a set of FIR filters that give us the hemodynamic response function for each individual feature. Typically in a motion energy model or a semantic model, there might be a couple thousand features. So we'll probably have something on the order of 4,000 to 5,000 features total that we're going to be fitting, and we fit them all simultaneously. So now each voxel generates predictions. So now we can take a new test data set, we can take the brain activity, and we can look at the predicted activity relative to what we expected to see, relative to what we can compare the predicted activity to the observed activity, and we can use that to validate our model. And this is going to give us, of course, not just statistical significance, but importance. It's going to tell us how much of the variance we can actually explain, and we can actually compute the noise ceiling on this. So one of the nice things about this is we can actually tell when we're done. When we're done is when we explain all of the variance in our data. Now, once we find some models that seem to work well, eventually we're going to either do that or we're going to get tired of playing this game. And then we can interpret the results. We can either look at individual receptive fields of voxels. A receptive field is a concept from neurophysiology. You look at an individual neuron and you try to see what it's tuned for. We can do that with voxels. We can look at all the voxels together in some feature space and try to recover the underlying structure of the feature space. Or we can look at the structure of the voxel features on the flat maps of the cortex and try to understand how the brain is representing this information. So in the low-level vision case, let's go through an example here. Our input here is movies. Our output is brain activity. So we take our movies. We nonlinearly map them into a bank of motion energy filters. Each motion energy filter is a cascade. We strip off color. Unfortunately, we don't have enough signal to noise to use color information in this experiment. We shove them through a bank of spatiotemporal Gabors. We take Gabors in quadrature phase. That gives us something which is analogous in primary visual cortex to something called complex cells, phase invariant grading selective cells. We put them through a compressive nonlinearity because it helps our predictions. And then we temporally downsample from the movie rate, which in this experiment was 15 hertz, to 1 hertz. And now each one of these 2,000 motion energy filters is used to fit a 6-tap FIR filter. And the linear sum of those gives us our bold response. And you might not think this works, but it actually works pretty well. These are flat maps of the right and left hemisphere. The visual system is in the middle here, and prediction is just given by the color. You can see that the predictions are quite good for this stupid motion energy model in primary visual cortex and other early visual areas. And other models that don't include, say, motion or orientation don't work as well. And if we look across the visual system, we see that in all of the early visual areas, these are just visual area 1, visual 2, 3, 3A, 3B, V4, all of these early visual areas and intermediate visual areas, this motion energy model works better than any other model we have. So this is a really good model, and it makes sense because we know that neurons in V1 are essentially Gabor filters, and this model is saying that a voxel, which is integrating from 1 million neurons, is taking a sum of many, many Gabor filters. You can look at an individual voxel. So this is the voxel's receptive field. This is the point in space that the voxel responds to. And you can see that it has spatial frequency and orientation tuning. You can put all these voxels together and recover retinotopic maps. Some of you may have seen this. The visual system, all the early areas are retinotopically organized. They have a spatial map, and you can recover this from motion, from movies. So that's pretty good. It works really well. It doesn't really tell us much we didn't already know. We already knew that primary visual cortex was a bank of Gabor filters, so it's not really all that enlightening. Let's go to the high-level visual system and see what we can do there. In this case, we are going to build a model that tries to extract information about parts of the visual system that respond to the semantic category of objects, like is there a dog, or is there a cat, or is there a person? So to do this, unfortunately, we can't label the movies by computer because computer vision really hasn't solved this problem yet. So we have to label them by hand. So we go through the movies, and we label them. Is there a dog? And if we know there's a dog, then it entails that there's a canine and an animal and so on. We know there's a woman, so there's also an adult. There's a person. In this clip, we know there's a jaguar, so there's a feline, an animal, and so on. This takes our movies from this pixel space into a feature space that's essentially just a giant matrix where the rows are 1,785 nouns and verbs, 1,785 most common nouns and verbs in the English language, and the columns are time. And this is just an indicator matrix of whether that noun and verb occurred at the movie at any point in time. So now we have our brain data, and for any given voxel, we've got some sort of temporal response. And we can use, again, ridge regression to estimate a finite impulse response filter that tells us how the brain, this one point in the brain, responds when any one of these 1,785 nouns and verbs appears in the movie. And this shows predictions of the motion energy model I showed you a second ago versus the structural model. This is early visual cortex, V1, V2. This is higher level visual cortex. And here, the orangey color indicates that the category model works best, and the bluish color indicates that the structural model works best. So you can see that there is a sort of continuous trade-off from this low-level model to this high-level model, both fitting well, which is just what we wanted, so that's good. We can go in and probe an individual voxel. So this is one individual 2-millimeter location in the brain. These are 1,785 nouns and verbs. These exist in a giant tree. This is a tree given to us by WordNet. The root of the tree is thing, and various kinds of things are root off the tree. So this is an organism, and a certain type of organism is a person, and there are different kinds of people like athletes and priests and firemen and stuff like that. Red balls here indicate semantic concepts that elicit a positive bold response, a positive blood flow response from this voxel, and blue is concepts that tend to suppress the voxel's responses. And you can see that this voxel prefers any kind of people. It also prefers any carnivores, any kind of people talking, any kind of ungulates, which are hooved animals, or rodents. Basically, this voxel responds whenever there's a face in the movies, and this makes sense because this voxel is actually located in a brain area that we already know about called the fusiform face area, which has a high concentration of neurons that are selective for faces. Now, we have a lot of data here. Each individual subject has 1,785 by 60,000 data points. That's way too much data to look at, so we have to do some sort of dimensionality reduction. Of course, the data here is all Gaussian, so the first dimensionality reduction you think of is principal components analysis. So we do principal components analysis on each individual brain, and we find out it takes about, say, 70 or 80 principal components to account for 95% of the data. That's essentially the data that we are given by the MRI machine, which is, of course, a limited device, right? It can only record so much. But if you compare the principal component solutions across individuals, you find that the first 5 principal components are almost exactly aligned across individuals. And only one of those is correlated with stimulus statistics. It's the first one. It happens to be correlated with stimulus energy. So let's look at this subspace, because this might give us information about how the brain is representing these semantic concepts. So in the upper left here is the first principal component. This essentially discriminates bright, fast things versus slow, dark things. And that's not very interesting because, of course, bright, fast things are going to elicit more neural activity, and that's going to elicit more blood flow. So that's not interesting. Let's throw that away. And let's look at the second, third, and fourth principal component. Now, to look at these data, what we can do is we can color each individual semantic concept by its loading on the second, third, and fourth principal component, and that's shown on the right here. This is the same tree you saw a moment ago, except now the color indicates the similarity of the semantic concepts from the point of view of the underlying brain representation. So yellow here is animals, green here is people, blue here is buildings, purple here is vehicles. You can see that all these groupings of semantic concepts tend to cohere, right? The brain is treating all the semantic concepts that occur at one branch of the tree. It's treating them essentially as an equivalence class. And that is, of course, what you would want for a system that is extracting semantic information from these movies. It's actually a little easier to see this in 3D. Remember, yellow here is animals, purple here is vehicles, blue here is buildings. This one branch that's sticking out and looks completely different from everything else, that turns out to be text. Text is represented completely differently from everything else in the brain. And you notice this green flat branch here is people. And you notice there's a small kind of orangey ball hanging out at the end of the people branch. That turns out to be athletes. For some reason, when your brain sees an athlete, it says, well, I'm not sure what that is. Is that an animal? Is it a person? I'm not really clear. So it's kind of represented halfway between. OK, so this is a really interesting semantic space. It tells you a lot about how the brain organizes information. But this actually, from a neurobiology point of view, wasn't what we set out to do. What we want to do is see how this information is mapped across the surface of cortex. And so now you can basically take the individual voxels, all 60,000 voxels that we measured, and you can project them into this three principal component space. And then you can color each voxel according to its loading in this principal component in this semantic space. And you can paint the surface of cortex according to this color scheme. So here, purple indicates sort of vehicles in motion. Yellow is animals and people. Blue is buildings, and so on. You can see that all over visual, higher order visual cortex, everything except this area called the temporal-parietal junction, all the way up to essentially the somatosensory strip, up into the precuneus and the retrosplenial cortex. Basically, all over the back of the brain, you end up with visual information of semantics. And you'd expect to see that. That's where the visual system is. It's in the back of the brain in humans. Now, these maps are interesting and very rich, but they're not stable, it turns out. And the reason has to do with attention. So the visual system is organized as a hierarchy, as I mentioned earlier. And neurons tend to pool information across many, many neurons at each successive stage of processing. So an individual neuron in V2 might have a spatial receptive field, the location in space that it can see, that's 4 times as large as the spatial receptive field in V1. So it's pooling over many, many V1 neurons. And this pooling continues throughout the brain. So if you have very tiny changes at one level of the visual system, those end up causing huge changes further on in the visual system. They can actually change tuning. So imagine that you have, say, a space here. We'll just have 2 dimensions, and that each voxel or each neuron is a basis function in this space, and they just tile the space. Now, you could also project the semantic concepts into this space, and you might have the semantic concepts arrayed like this. Now, if neural tuning changes occur at a high level of the visual system, what that's going to do is it's going to actually shift voxels or neurons over toward different semantic concepts. So for example, if we ask people to attend to humans, all the neurons and many of the neurons and voxels might shift over and try to represent that part of the semantic space better, and leaving this part of the semantic space less well sampled. And then if we ask the person or the system to attend to vehicles, then the neurons or voxels would shift up towards vehicles, overrepresenting this part of the semantic space and leaving this part of the semantic space underrepresented. And this is a sort of a matched filter mechanism, right? If you have fixed hardware, this might be an optimal way to try to expand some part of the semantic space at the expense of other parts. And it turns out this actually happens. This is one individual voxel when people are watching the movies and we ask them pay attention to humans or pay attention to vehicles. You can see when they pay attention to humans, we get bold activity elicited by faces of people and animals. But when they pay attention to vehicles, although we still get face-related activity, we also now end up with a bunch of activity related to vehicles that we didn't have before. So the tuning curves are actually shifting. And this actually has an effect on the maps. So here is a semantic map for passive viewing when people aren't attending to anything. Again, yellow, green here is humans and animals. Red, purple is vehicles in motion. And when people are attending to humans down here in the lower left, you can see that the map becomes much more green-yellow. And the reason for this is that humans are now overrepresented. And when you pay attention to vehicles, the map all of a sudden becomes very purple because now vehicles are overrepresented. So voxels are shifting their tuning curves all over the brain when the attention is changing. And this is very consistent with what we see in recordings from single neurons as well. Single neurons also shift their tuning curves as attention changes. So this makes the job of trying to figure out the brain a bit more complicated because now this is a very internal state-driven kind of representation. And if we want to come up with a general model for how the brain's representing information, we have to put mental state in as a sort of a conditioning variable or a hyperparameter, and that makes everything much more complicated. Is this time accurate? I doubt it. Really? Huh, remarkable. Okay, so I'm going to, since I have a bit of time, I'm going to discuss one more model that is a scene category model. And the reason I'm going to bring this model up is that this model is trying to take advantage of the natural scene statistics. So in the previous two models I showed you, the models were essentially created by hand. When we had to develop a motion energy pyramid, we just made some arbitrary decisions about what the temporal and spatial frequency bandpass of the visual system was and how much information we could recover from MRI, and we designed a tailored motion energy pyramid for the early visual system. In the case of semantic categories, we just used what WordNet gave us. WordNet is essentially an is-a hierarchy that's a tree drawn from the English language, and because WordNet is taken from language and not vision, it doesn't necessarily form a good representation of vision. So in this situation, what we wanted to do was try to discover if there were brain areas that were selective for scenes. These have been reported before in MRI experiments and in neurophysiology experiments in animals. But we didn't want to pick an arbitrary set of scenes. What we wanted to do was use the natural set of scenes given to us by images. So we took a large ensemble of images, and we labeled all of the objects in the images by hand. And then we gave them to an algorithm called latent Dirichlet allocation. LDA, for those of you who know, it's actually a topic model that was developed for text retrieval. The idea is you have a whole bunch of, in this case, scenes that all have different kinds of objects in them. But underlying this distribution of individual scenes are latent scene categories that essentially have a typical probability distribution for the objects that they can contain. And the cool thing about LDA is it allows an individual sample scene to be a mixture of multiple scene categories. So the first thing we do is we take the individual labeled objects from thousands and thousands of scenes, we give them to LDA, and it returns a bunch of categorical features that are essentially the latent categorical features of the scenes. And then we're going to use those as a feature space, and we're going to fit model weights to each and every voxel, and then we're going to get predicted voxel responses. We can also use this in a decoding mode that I'll get to at the end of the talk. So these are the latent scene categories we get out of our huge ensemble of natural images. And of course, LDA doesn't actually label the categories. It just gives us the probability distributions of the words in the categories. But you can see that these categories kind of make sense. There's a category fish, rocks, water, bushes, man, sand, seal, rock, boulder, planks, shark. So this category might be, for example, a beach category, right? And you can go through and label all the categories. You can have as many categories as you want in LDA, from 1 to infinity. We can't recover infinity. Your brain, might encode 50,000 categories for all we know. But we're not going to get that much information out of MRI. We're going to get some limited number of categories because MRI is a signal-limited method, right? We've got 60,000 voxels, not 10 billion neurons. So it turns out that the human brain, we can recover about 25 categories from the brain using this model. And when we look at the predictions of this model, you see that it fits really well in this weird kind of patchy arc. And this patchy arc is actually the higher level semantic areas that surround primary retinotopic visual areas. This model fits pretty well. It fits as well as the object model using WordNet that I showed you a minute ago. But this model is a scene category model, not an individual object model. And it was recovered directly from natural image statistics. So the conclusion here is that the human brain seems to accurately represent the distribution of latent scene categories in natural scenes. This just shows individual tuning for individual areas like PPA, the parahippocampal place area. You can see that the parahippocampal place area in all of the subjects responds more when you get an ocean or freshwater or a highway or a boatway or an urban street scene. It responds poorly to plants or people. And this is essentially, for all of the visual areas that we have some vague idea for what their category selectivity should be, this model correctly recovers it. And it also recovers information in a lot of areas that we never knew about before. Now, you may have noticed that I skipped over mid-level vision. And I did that for a reason. Mid-level vision is very frustrating. It's frustrating for us in neuroscience, and it's frustrating for folks in computer vision, right? Computer vision works really well for taking individual low-level pixel representations and mapping them into, say, some sort of pyramid structure and then extracting local edges. That works really, really well. And once you have essentially a labeled scene or a scene where the objects, excuse me, not labeled scene, but a scene where the objects have been segmented, then there's a lot of classifier-based methods in computer vision that can recover the individual classes and can do all sorts of interesting things. The problem for computer vision is when you have a complicated scene and it has not been segmented. That is essentially a sticking point. It's I call this the mid-level vision problem, which is essentially a problem of segmentation and grouping. It turns out we're in exactly the same bad situation in human vision. We have really good models of early visual system. We have a pretty good understanding, we think, of how higher level vision works. But this intermediate step between the low-level features mapping from motion energy filters up to semantics, we just don't have any good model for that. We don't have a good model for that in animals. The models for neurons in visual area 4, for example, in non-human primates are horrible. They don't work at all. We don't have a good model for this in human MRI. In fact, until recently, the best model we had for mid-level vision in humans was just using the Gabor wavelet pyramid, and we know that's the wrong model, right? The Gabor wavelet pyramid, the motion energy pyramid, is the correct model for V1. There's no reason to take that same exact information and keep re-representing it over and over again. Instead, what should happen is these higher areas should be integrating information from earlier areas and information from higher areas and essentially non-linearly transforming it into a new feature space to make new kinds of features explicit so they can be easily read out, because all of these maps are essentially just different explicit representations of intermediate and high-level features. So the question is, what are these mid-level features? That's our problem. To try to get at this, this is, sorry, an illustration I should have shown. This is a picture. We can get the luminance edges out. We can get the labels out. If they're given to us, we can build a system that processes that. But moving from the luminance edges to the semantic categories is where the problem is. So to try to get this issue, we created artificial movies using Blender, which is an open source animation software. And the nice thing about Blender is now we have ground truth. We have a depth map for all the pixels. We have all the normals. We have the segmented objects. And now we can use these different kinds of features to try to model our system. So we can take segmented images. We can come up with sort of an object blob model. We can take the segmented images and the normals and come up with all the individual edges of all the objects in the scene. Or we can use a more simple representation that only recovers the silhouette edges of the objects in the scene, but discards all the intermediate middle features. And when we do that, we find out, shown as up here on the left, that the object model works poorly in all visual areas. The all scene contour model works really, really well in V1 and V2. But this object silhouette model, which, if I can go back, is this model here. This object silhouette model works really well in higher level visual areas, V4 and LO. These are intermediate visual areas. And what we think is happening here, you can see the change up here. The early visual system in red is where the object contour model works well. And the higher order visual areas in blue are where this silhouette model works well. So we think this is a reflection of the figure ground segmentation operation that occurs, we all think, in intermediate vision. And we're sort of seeing an echo of that in the MRI data. So I'm pleased that this has gotten us somewhere. This is the first time we've ever had a model that actually works better than the Gabor wavelet pyramid model in these higher order visual areas like V4, these intermediate visual areas. But this isn't done yet. I've seen neurophysiology data from area V4, and the neurons are hideously complicated, and how they're implementing some sort of object segmentation and grouping process is still largely mysterious. So now in the last part of the talk, I'd like to switch to decoding. I should mention that I'm not a big fan of decoding. and that's because I don't view it as a science problem. Decoding is an engineering problem. It's a reverse engine. It's an engineering problem. So in encoding, we have a world here, and we have some brain activity that is elicited by the world. And to model that, we can take our stimulus, and we can nonlinearly map it into some feature space. And then once we have our feature transformation, we can do linear mapping onto our brain activity data. Bold is blood oxygen level dependent response. To do decoding, we can simply take all of these individual bold responses, we can linearly map them back into the feature space, and then we can just decode the features. Or if this nonlinear transformation is invertible, we can decode back into the stimulus space. Now, this is a perfectly fine thing to do if you want to build a piece of equipment to do brain decoding, but it's not an interesting scientific problem. And the reason it's not interesting from a scientific point of view is you can always convert an encoding model into a decoding model just by Bayes' theorem. So in Bayes' theorem, this modeling framework turns into the following equation. The model gives us a likelihood of the response given some function of the stimulus, a nonlinear function of the stimulus. And if we multiply that times a prior, which is just the prior probability of some function of the stimulus, we will get a posterior, which gives us some function of the stimulus given the response. So whenever we have an encoding model, we can always convert it to a decoding model. And of course, if we have a decoding model, we can always convert it to an encoding model. So really, once you build an encoder, the job's kind of done. You get the decoder for free. Now, there's a whole bunch of applied math tricks that have to do with how you actually implement this thing that are kind of annoying. But that's just time. You know, it's not kind of an intellectually interesting endeavor. It's just you just throw everything you can at it till you get it to work, and you buy more computers. OK, so let's look at some examples here. This is an example for static images. This is an older study, but it's a good place to start. On the top here are 3 images that we showed people in the magnet. This is a harbor scene, and the reconstruction here you notice is a harbor scene. Here's a bunch of people in a room. Here's a bunch of people in a room with their hands up in the air. And then here are 2 buildings in the front and a building in the back. And our reconstruction says that there's 2 buildings in the front and a building in the back, although one of these buildings is a Coliseum. Now, you might ask, why is it picking these specific images? Why didn't it find these images? Well, this has to do with the nature of the prior. Ideally, in doing decoding, we would take all of our images, we would shove them through the models and get our likelihoods, and then we would multiply them times the correct natural image prior. But all of you in the audience know we actually don't know what the natural image prior is. We don't know what the prior is on natural images. We don't know what the prior is on natural movies. This is, of course, a core problem in computer vision, but it just takes too long to estimate this with current computers, so nobody bothers, right? So we don't have a prior. So what are we going to do for a prior? Well, one of my graduate students came up with a brilliant idea. He said, look, we don't really need the actual prior. Let's just use an empirical prior. So we went out to our friends at Flickr and said, can you give us images? And they gave us a hard disk with 50 million Flickr images on it, and that's our prior. In Bayes' world, this is called a stick prior, right? The probability that we can reconstruct any image that's actually on this Flickr hard disk is 1 over the number of images, and the probability that we can reconstruct any other image in the world is 0. But obviously, as the prior grows larger and larger, if Flickr had given us 5 billion images rather than 50 million, we would have better reconstructions. So really, these reconstructions are the best image we could find given the brain activity elicited by this image and our models in the database of Flickr images that we had. And you notice these are pretty close. We've lost the contrast here. This is black and this is white. But that's OK, because the visual system actually has 2 channels, one for luminances below the mean luminance, and one for luminances above the mean luminance. And in V1, at the single neuron level, you can tell these apart. But in MRI, they're all mixed together. So we lose contrast. So that's fine. Altogether, these reconstructions are actually pretty decent. Now, MRI is very slow because it's measuring blood flow. It takes actually about a second, 2 seconds for the MRI signal to manifest. It peaks at about 4 seconds, and it doesn't decline to 0 until about 10 seconds. So it's very slow. It's actually 1,000 times slower than neurons, 10,000 times, because a neuron can fire up to 100 times a second. So it's always been an open question, can you decode movies with this technology? And in some sense, you shouldn't be able to, because we're only sampling the brain once every couple seconds, and the blood flow signals are very slow. But we decided to try this. We took the movies. These are the movies up here at the upper left that we presented. We used our motion energy model, and we tried to decode brain activity from early visual cortex. On the bottom right here is a solution that a lot of people jump to as their first decoding solution. It's called direct decoding, and you can see that that works very, very poorly. On the upper right is the maximum of the posterior for the images. So this is our best guess as to the decoded image. And you can see that sometimes this works really well. If this is a person, that works well. If this is something abstract or rare like an elephant, this works poorly. And that's because our prior, in this case, was actually 5,000 hours of video downloaded from YouTube. Google wouldn't give us images, so we, I don't know if stealing is the right word. We generated random URLs, and for 6 weeks we just pounded Google until it gave up its goodies off of YouTube. They were completely random. 5,000 hours of video. We don't know what we got. So 5,000 hours turns out to be about the amount of time you're awake during 1 year. So you can think of this as sort of 1 year of your personal visual experience if all you did during that year was watch YouTube. So there's a lot of people on YouTube, and so we can reconstruct people really well. But if it's something abstract, we actually can't reconstruct it very well. And in that case, instead of just taking the peak of the posterior, you can average the top 100 things in the posterior, and you get this thing down here called the average height posterior. And you can see this kind of reconstructs the motion energy in the stimulus, even though it's hard to tell exactly what the stimulus is. Both of these are valid reconstructions. Now we've also done decoding using this semantic model that I told you earlier about. This uses WordNet. So here we're simultaneously decoding 1,785 nouns and verbs from the brain. This uses these higher level semantically organized visual areas. I've just arrayed the words on the first two principal components here so they don't overwrite one another. And the size here is the probability that that thing appears in the movie. You can see that sometimes things will appear slightly before they actually appear in the movie. That's not because our subjects have ESP. It's because we slightly overcorrected for the hemodynamic delay. So here we are on Waikiki Beach. You can see we get building, body of water, city, walk, people, tree, sky, road. There's no mountain, but that's OK. OK, this is quick, so we lose it. But then we get text here for all bets are off. Now we have 3 women, so we get talk, woman. There's the man. You notice he came up in a bit late. Talking, flower, lots of talking. Now we switch to an ocean scene. We get fish, swim, water, body of water, ocean floor. This is a manatee. We don't have manatee. It gets whale. I think that's pretty good, actually. In general, this semantic decoder works pretty well. I mean, it's really remarkable to me. You can take this really kind of clunky MRI machine, which is a very beautiful piece of physics, but is measuring something that's barely related to neurons in the brain, and you can recover this kind of information. And of course, that's because there's a lot of information in your brain, right? But if you do a back-of-the-envelope calculation, one of these sort of physics, assume a spherical calculations, at best, MRI is recovering 1/1,000,000th of the information in the brain at any given point in time. So it's a really sparse sampling of what's in the brain. Here's the LDA, the latent Dirichlet allocation model that I showed you earlier. This used static images rather than movies. So here's a guy with a tarantula on his face. This was not used to fit the model, right? We're doing full cross-validation here, so the model never saw this guy. But it recovers the likely scene. It says, well, I think this is land animals, and there are few people category, and maybe the food category, I'm not sure. And then it says likely objects are man, woman, person, head, animal. There's a sporting scene. It says this is a sporting event and a large crowds and a few people scene, and it's got an athlete and people and man. And you can see in all these cases, the LDA model also recovers information really, really well. So this is, I find it both exciting and very, very scary that we can recover this much information in this day and age using MRI. And I would argue that this is essentially early days for brain decoding. This is my analogy. This is the very first photograph. Many of you have probably seen this before by Joseph Nietzsche in the south of France in 1825, taken out his farmhouse window. You can kind of see there's a building here, maybe a roof and a building, another building over here, and maybe some sort of tower here. This is a really bad photograph, but it's obvious that it's a photograph, right? You can tell what the things are in this photograph. And I would argue, currently, this is where we are in brain decoding. Now, it's 200 years later, almost, from this photograph, and now we can do photography so well that no one even wants to pay for it anymore. It's basically free. And eventually that's going to happen with brain decoding as well, because remember, there is an intimate link between encoding models and decoding models. So if you understand how the brain works, if you as a neuroscientist can say, I can accurately predict what the brain will do in situation X, you can decode the brain. These two things cannot be unlinked. One of them always gives you the other. So that means that brain decoding is going to proceed, and eventually we're all going to be wearing thinking hats. Instead of Google Glass, you'll have Google Hat, and you'll say, Google, search something for me, and it'll just transmit to Google directly out of your brain, which I find incredibly scary. So especially in this country, which has essentially a very poor record of privacy rights. I think this is something that we as neuroscientists have to worry about now because this is going to come down the pike. We don't know when. We don't know what technology it's going to be. It's not going to be an MRI machine, but it's going to happen. So if you're interested in playing with some of this data online, we have an online brain viewer at my website, gallantlab.org, and you can click on parts of the brain. It'll show you what they're tuned for. You can click on the semantic tree. It'll show you what parts of the brain like those semantic categories. It won't run on your phone because it's too beefy. It uses a lot of really nasty JavaScript and HTML5, but it will run on your laptop. And also, if you want to play with this data, you can. All of our data is up at crcns.org, which is a data sharing site for physiology and MRI data. You can download all the data you want. And I'd just like to acknowledge the people who did this work. Alex Huth is a fantastic graduate student of mine. Mark Lesquois, I didn't have another picture of him, is the guy that did the mid-level vision work. Alex did the semantic tree. Dustin Stansbury did the latent Dirichlet allocation. And Tolga Chukar did the attention work. Thank you very much for your time. You've been great.